Hello, everyone. The study that I chose is the role of JNK and p53 in the regulation of secretory function and neuroglial cells of various types of beta amyloid-induced neurodegenerations. So before we get into the study, I would like to talk about the key information of this study, which is what we're going to be talking about throughout the whole presentation. First of all, Alzheimer's disease. As many of you know, or as everyone knows, Alzheimer's disease is a neurodegenerative disorder that is characterized by progressive decline in cognitive function and memory. It is the most common cause of dementia, and it affects millions of people worldwide. The disease is characterized by the accumulation of abnormal proteins deposits in the brain, which include the beta amyloid plaques. This leads to the death of the nerve cells and the disruption of neural networks. As the disease progresses, individuals experience worsening of memory loss, confusion, difficulty with language and communication, impaired judgment, and behavior and personality changes. Alzheimer's disease has a significant impact on daily functioning, and even though it is so common, it has many research into it. There has yet been found a cure or a preventative solution to it. Second of all, I would like to talk about neural stem cells. NSCs are a type of stem cells that are found in the nervous system, and they have the remarkable ability to differentiate into various types of nerve cells, including neurons, astrocytes, and oligodendrocytes. They are primarily located in specific regions of the brain, such as the subventricular zone and the hippocampus. NSCs play a crucial role in the development repair, and the maintenance of nervous system throughout life. A key characteristic of NSCs is that they include self-renewal capacity. This allows them to produce identical copies of themselves, and their ability to differentiate into different cell types depending on the signal they receive from the environment that they are in. This plasticity makes NSCs a promising target for like generative medicine approaches aimed to treat in neurodegenerative diseases. In this study, we specifically talk about Alzheimer's disease. Now, beta amyloid is a peptide that plays a central role in pathogenesis of Alzheimer's disease. It is derived from the cleavage of a larger protein called amyloid precursor protein by enzymes called beta and gamma secretase. In Alzheimer's disease, beta amyloid peptides aggregate to form insoluble plaques, which accumulate between nerve cells in the brain, and they are believed to contribute to neural dysfunctioning and cell death, leading to the characteristic cognitive decline and memory impairment seen in this disease. Now, another thing that I'd like to talk about, and it's not seen in the slides, is JNK and p53, which are mentioned in the title. JNK is a type of protein kinase involved in cellular signaling pathways that regulate various cellular processes. This includes cell proliferation, apoptosis, and stress responses. JNK is activated in response to various stressors such as DNA damage, oxidative stress, and anti-inflammatory cytokines. Once activated, JNK phosphorylates target proteins, leading to changes in gene expression and cellular behavior. In the context of neurodegenerative diseases like Alzheimer's, JNK signaling has been implicated in neuronal death, inflammation, and synaptic dysfunction. Now, p53 is a tumor suppressor protein that plays a critical role in regulating cell growth and preventing cancer. It is often referred to as the guardian of the genome due to its ability to monitor the integrity of DNA and initiate cellular response to DNA damage or other stressors. Now, in the context of neurodegenerative diseases such as Alzheimer's. P53 may play a role in regulating neuroglial cell function and the production of neurotrophic growth factors, as described in the study. Now, the goals of the study. Um, this study aims to investigate the role of two signaling molecules, which were mentioned and explained before, that are JNK and p53, in the production of neurotrophic growth factors such as astrocyte, oligodendrocytes, and microglial cells in an in vitro model of beta amyloid-induced neurodegeneration, which mimics Alzheimer's disease. By understanding how these molecules influence the secretion of factors, the research seeks to shed light on the mechanism of neurodegeneration in Alzheimer's disease and explore new avenues for potential therapy targeting these pathways to stimulate neurogenesis and tissue repair. Moving to the approach that they had, like the approach to the goal. Um, the authors of the research aim to investigate the role of two signaling molecules, as it was mentioned before. And their approach was by number one, experimental model. They utilized an in vitro model of beta amyloid-induced neurodegeneration, which involved exposing cultured neural cells to beta amyloid to stimulate the pathological um, conditions observed in Alzheimer's disease. Sorry. Number two, cell culture. The authors isolated astrocytes, oligodendrocytes, and microglial cells from subventricle zone from the subventricle zone of mouse brain tissue. These cells were chosen because of their involvement in neuroinflammation and neuroprotection, which are relevant processes in Alzheimer's disease pathology. Number three, treatment. The isolated cells were treated with beta amyloid to induce neurodegenerative conditions in vitro. This treatment mimicked the deposition of beta amyloid plaques observed in the brain of individuals with Alzheimer's. Number four, inhibitor testing. The authors applied inhibitors of JNK and p53 to the culture cells to assess their effects of secretion of neurotrophic growth factors. These inhibitors were used to selectively block the activity of JNK and p53 signaling pathways. Number five, assessment. They measured the secretion of neurotrophic growth factors and assessed neurosphere formation as indicator of neurogenesis. This allowed them to evaluate effects of JNK and p53 inhibition on the cell's ability to produce factors that promote growth, um, growth and survival. Number six, statistics analysis. The results were analyzed using appropriate statistical methods to determine the significance of differences between the experimental groups. Number seven, interpretation. The authors interpreted the findings and to understand how JNK and p53 signaling pathways regulate the production of neurotrophic growth factors by neuroglial cells under condition of beta amyloid-induced neurodegeneration. Number eight, implications. Based on their results, the authors discussed that potential therapeutic implications of JNK and p53 signaling pathways to promote neurogenesis and tissue repair in Alzheimer's disease. Overall, the authors employed a systematic approach combining the cell culture techniques, pharmacological interventions, and biochemical assays to investigate the role of JNK and p53. Now, the major results, the increased secretion, effect of inhibitors, stimulation of neurogenesis, and potential therapeutic implications. The study reveals several significant findings. First of all, exposure to beta amyloid led to increased secretion of neurotrophic growth factors by astrocytes and microglia cells, but not by oligodendrocytes. Secondly, inhibiting JNK did not affect astrocyte function, while blocking p53 decreased their secretion of neurotrophic factors. However, both JNK and p53 inhibitors increase the secretion of neurotrophic factors by oligodendrocytes and microglia cells. Thirdly, inhibition of JNK and p53 promoted neurogenesis by enhancing the growth potential of nervous tissue progenitors. Sorry. These results suggest that targeting JNK and p53 signaling pathways could hold promise as a therapeutic strategy to stimulate neurogenesis and promote tissue repair in Alzheimer's disease. Now, for impact and future research, this study provides a valuable insight into the role of JNK and p53. So, for me, it made me understand how JNK and p53 can play a role not only on Alzheimer's disease, but neurologically, it can have a big impact in tissue repair and neuroprotection. For future research, further investigate other areas where JNK and p53 could have been involved or could be involved. Maybe explore the efficiency and safety of JNK and p53 inhibitors as potential therapeutic agents in preclinical and clinical studies. So by addressing these areas, future research can build upon findings of this study and advance our understanding of Alzheimer's disease, but the pathogenesis and potential therapeutic information. So for takeaways, my takeaway is in this course we learn a lot about cell, the basics, like the whole structure of a cell and how one cell can affect the functioning of many other cells or many other parts or systems in our bodies. While I was reading the research, I knew a lot of things. I knew about p53 in the context of cancer, for example, but now I can apply it to the context of Alzheimer's, and I'm interested in investigating more about p53 in that context, or JNK and maybe other things that were mentioned here, such as the enzymes and others. More so, I come from a family that we've had many members with Alzheimer's disease, and growing up, I did not quite understand the role they had or how it actually functioned. So this study helped me understand way more, not only Alzheimer's, but neurodegenerative diseases in general. Thank you.